Good morning, or afternoon, or evening from wherever you are. And I hope you and your families and communities are doing as well as possible in these strange times. What I want to do is take about 12 minutes to talk about the ability of corals to adapt to climate change, especially ocean warming, and whether or not the intrinsic variation in their ability to deal with temperature allows an opportunity to use that evolutionary potential in helping inject higher resilience into reefs in the future. And these considerations sort of stem from this kind of observation, very common out on reefs where there's a natural bleaching event where some corals, like on the left, don't bleach, whereas other corals of the same species, like on the right, do bleach. And that gives us the idea that there is variation within species in how corals deal with the external environment, and maybe that provides the fuel for evolutionary adaptation into the future. Well, there's a whole raft of theories out there, as far back as Levine in 1953, that lay out the conditions under which we might expect there to be variation within populations, as genetic polymorphism for ability to deal with differences in the environment. And these theories lay out those conditions, which essentially say if there are different alleles within the population and those alleles work better in some environments than others, that is, if there's a trade-off and there's dispersal between habitats, then oftentimes those are conditions under which the polymorphism is maintained in these populations. And models have taken these kinds of theories and the polymorphisms that are expected from them and projected that into the future, like this one from Rachel Bay in 2017. And here a southern Pacific population of corals is imagined to be evolving over the next century under different levels of climate change. And when climate change is relatively mild, like the blue lines, that population can persist and adapt as the temperature slowly warms. Whereas under very strong examples of climate change, like RCP 8.5, populations evolve for a little bit, fall behind, and then collapse. This is Tim Walsworth's model, 2019. With no genetic variance, coral populations collapse. With a little bit of genetic variation, they retreat a bit, but they still can maintain. This is a recent model by Lisa McManus. Here, V is the variance of the population. Where it's low or zero, then the coral populations tend to collapse, whereas if it's even small, like V equals 0.01, then those populations do much better. And then just a few weeks ago, Cheryl Logan and colleagues took those models and applied them to the symbionts as well, not just the coral host. So these theories suggest that we might actually expect to find polymorphisms within populations under some conditions to respond to climate change. And the model suggests that under some conditions that might be enough to spur evolution in the future so that adaptation can increase. But what do we really find in the field? Well, we've worked in American Samoa for a number of years looking for adaptive variation, particularly in heat resistance. And in this case, two pools, back reef pools, a warm one and a cool one. And what we found is that there is difference in the ability of corals to withstand bleaching events based on whether they live in the warmer pool or the cooler pool. And about 50% of that is likely to be due to adaptive loci, whereas the other 50% is likely to be due to acclimation. Other studies have been looking similarly and finding similar kinds of patterns. Dixon et al., for example, did a breeding trial of corals on the Great Barrier Reef from north and south. Their hybrid offspring were intermediate in their heat tolerance, suggesting that this was a multi-locus trait controlled by many, many loci. And more recently, Zach Fuller and his colleagues in New York and Australia looked at variation in bleaching across a natural bleaching event, correlated that with genome-wide SNPs, and found that about 2 or 3% of the bleaching variance within those corals was due or ascribable to the genomic SNPs. Well, in order to expand this kind of work and go beyond, in our case, just a couple of populations, we've been working at the Palau International Coral Reef Center with researchers there like Victor Nestor and the director Yim Gulbu'u, And they have been helping us collect corals from a wide variety of different reefs around Pickwick, bring them back to the lab, and then do standardized bleaching tests of these corals in order to be able to compare their heat tolerance in a variety of different heat regimes. And this is sort of the setup we have there. And just to um, jump to the results, each of the red dots that you're going to see here represent a heat-resistant coral among the 400 that we have tested across 38 reefs in Palau. And two things sort of come off of this map. One is that these corals are pretty widespread and common. We find them in most of the reefs that we have examined populations, and even some of the four-reef habitats. But there are differences among reefs where some reef complexes have more heat-tolerant corals than others. For example, the corals, reefs that are surrounded by red here are warmer, and they also have a higher fraction of heat-resistant colonies than the corals, for example, surrounded by the blue square here. Those are in general cooler, and they have a lower fraction of heat-tolerant corals. But we also find that there's other features associated with corals that have turned up in our studies to be pretty important. This is one, for example, and that's the symbiont load in these coral populations. On the left is an individual of Acropora hyacinthus, the tabletop coral that we have been studying, sort of chocolate brown, high number of symbionts per coral cell, whereas on the right, same species, on a different reef, much paler in color, so pale you can see the underlying color of the skeleton. Um, and that difference in symbiont load is a striking feature of different corals on the same reef sometimes and on different reefs. And you can see that here in this histogram, looking at the blue bars. This is the fraction of the cells in a particular branch that are symbionts. These are assayed by stripping the tissue and then running it through a FACS cell sorter. And that fraction of symbionts ranges down at the low point to 4, 5, 6% and upwards of 20% or even larger. And that very big difference in the number of symbionts per coral, then it turns out, is also correlated with their bleaching resistance. On the y-axis here is the amount of chlorophyll that's left in these colonies after our standardized heat testing. The higher the number, the higher the bleaching resistance. And corals with high bleaching resistance on the left of this graph have generally start out with a lower symbiont load than corals that have pretty low bleaching resistance and start out with a much higher symbiont load on the right. And this relationship, this negative relationship between bleaching resistance and symbiont load, was actually discovered by Ross Cunning and Andrew Baker's lab some time ago. And they ascribed this potentially to the negative consequences of having too many symbionts in the coral tissue. But if you think about it, symbiont is actually a pretty strong feature of the lifestyle of reef building corals. In particular, their role in coral nutrition is pretty strong. We were able in our study to go back to the same corals that we had originally sampled. Each of them is mapped and tagged. We went back after a year and photographed them again. Comparing photographs, we could get an idea of the measurement of the growth of each of these individual corals. And here, if you look at annual growth of these corals, you see that the higher the symbiont load, the higher the growth rate. Low growth rates are generally associated with relatively low symbiont loads in our study. In both these graphs, there's a lot of scatter, there's a lot of variation between them, but the trends are significant. They suggest that there's actually a trade-off between growth and retention that is driven by symbiont load. Bleaching may actually be part of a four-way trait that involves the intrinsic heat resistance, involves the symbiont load, the symbiont type, and the growth rate. All of those are traits that are interdigitated together, and then doing a restoration project that only looks at one of those traits, that is heat resistance, may not actually be the best way to go about it. For example, if you take the corals with the highest bleaching resistance, they might have some of the lowest growth rates. So our work in Palau is ongoing, and what we have been doing is taking these kinds of results and then taking them to the next level. All the results I've been telling you so far have been results from field collected colonies, and we're currently in the process of doing the common garden experiments that can show how much of the difference there is between habitats is actually due to intrinsic versus acclimatory differences. Katrina Armstrong has a poster on this that I encourage you to go see with the preliminary results of the experiments that we have laid out there. That also allows us to look at the symbionts involved in these particular corals. We know from our prior study that the vast majority of the corals in our sample are dominated by cladocopium, but there's lots of different traits of cladocopium. There's lots of different varieties. Brendan Cornwell is giving a talk about the genetics of coral symbiont coevolution in this and other systems based upon a new full chromosome length sequence, a genome sequence of Acropora hyacinthus that we have just produced, in this case comparing it to Acropora mellipora. So see Brendan's talk to hear about how you can take this kind of genomic data and the genomic data that come from the intrinsic symbionts in each of the corals and then look for correlations among them. We're also looking at the next stage in bleaching. That is, not all corals die when they bleach. Recovery dynamics is another really important part of how corals react to different environments. And here Neil Walker has been doing a set of recovery experiments with the same colonies that we have resistance phenotype data for. All across what we've been trying to do, and so many of the rest of the people in this meeting and in coral work all over the world, is to take the deep new science in coral biology and apply it to communities out in the field, trying to add resilience to coral reefs around the world, trying to set us up so that coral reefs in the Pacific and the Caribbean and everywhere have a better chance of surviving the coming climate crisis, and then can begin to grow back when that crisis abates. Thanks very much.